Good afternoon. Thank you for coming to my presentation. I'm Jyoti Rangineni with Fluid Air. So today we'll be discussing about how a continuous drying process, electrostatic drying, can help with maintaining stability of thermosensitive molecules and materials during drying, but remove the bottleneck traditional and conventional drying processes such as lyophilization can create during manufacturing processes. So some basics with electrostatic drying. Electrostatic drying basically originated from spray drying, but it is a temperature sensitive drying of liquid materials to produce powders. We introduced the unique electrostatic effects during the drying process that allows for non-destructive and efficient drying, at the same time efficient drying to produce dry powders. So you have all the principles of spray drying, but with the unique concepts that the electrostatics introduces into the drying process. So a quick comparison between freeze drying or lyophilization and the conventional spray drying. What you see in the green bar there are the outlet temperatures. As I said, the technology does originate from spray drying. So the green bar shows the outlet temperature, or what the dry powders do experience at the exit, the dry powders experiencing exposures to the temperatures. So between 35 to 100 C. I'll explain more in the next slides what that means compared to the very high temperatures in conventional spray drying and, as you know, the core temperatures of the lyophilization. So very quickly, if you're not familiar with spray drying process, what you see there is a production scale machine of electrostatic drying. The liquid feedstock is introduced at the top via a two-fluid nozzle. The liquid feedstock is atomized into fine droplets. At that point is also where the electrostatics is introduced or dispersed. A charge is dispersed onto the droplets. These droplets are flown into the drying chamber through a stream of drying gas that enters the chamber in a tangential flow that floats all those droplets in a cyclonic fashion through the drying chamber. And as the droplets float through the drying chamber, they're drying into dry powder particles, which then flow into the separation plenum, which houses our filters. The filters now separate the powders and the very wet gas vapor. The powder is now falls into the collection cone and the collection jars, or whichever mechanism you're trying to collect. And the wet gas is now passed through to a dehumidification unit and recirculated back into gas heaters and recirculated back into the drying chamber. So the drying gas in all of for electrostatic drying is always nitrogen gas. Because of the electrostatics, it's a hazard. So all of the electrostatic drying units operate with nitrogen gas, and the operating temperatures are between 40 to 140 C always. They do not go higher than that. So the principles of the technology, what happens when electrostatics is introduced into the spray drying process? Multiple phenomena occur. So in no particular order, in no particular sequence and order of importance, the four phenomena that are introduced are electrostatic induction, Coulombic forces, modification of the spray trajectory, and an electric field that is developed because of the charged particles. Because of these four phenomena, a new drying kinetic is introduced. The new drying kinetic is that there is an increase in evaporation rate at lower temperatures. Because of reduction in surface tension, increase in surface area for heat transfer. There is an enhanced protection of active components, and because of the protection, there is low thermal distress. This is going to be the theme of the drawing. There is protection of the solids or your thermosensitive materials, your peptides, oligonucleotides, or anything that the formulation requires. You're trying to stabilize within your formulation. So, of the 4 phenomena, I'll try to go through how the phenomena contribute to the drying process. Electrostatic induction. When the electrostatics is introduced, what happens is there is a charge dispensed onto the droplets as they are formed. The charge, what it does is there is a redistribution of electric charges within the droplet. There is a redistribution of those charges in the droplet. And because of this, there is an agitation within the liquid molecules in the droplet. And because of the mobility of those molecules, there is an increase of migration of liquid. When the droplet surface, the liquid molecules on the droplet surface are evaporating. Because of change in surface tension, there is movement of liquid molecules to the surface of the droplet. Now, increase in agitation increases that motion. That increases the rate of evaporation. And since the charge is now on the surface of the droplet, based on the affinity of the components within the droplet, you're also now seeing a layering or a stratification of the components within the droplet. I want you all to hold on to that thought because I have some case studies based on that I can show you, demonstrate to you what that effect can mean on the particle after drying. The next phenomena is spray trajectory modification. So when the droplets are atomized in conventional spray drying, it's a conical spray. But when the droplets are charged, now you have charged droplets. There's repulsion and attraction among the droplets. Because of that, the trajectory is no longer conical. What you're seeing is more dispersed. What this means is the droplets are much further away. There is more drying gas between the droplets, more chances of heat transfer. The 2 other phenomena, Coulombic forces and electric field. What Coulombic forces does is it increases motion of those droplets, meaning that what you see around the droplet for a suspended droplet is that wet vapor, saturated vapor, where the droplet is giving off drying, and that wet vapor that forms a protective evaporative cooling. A moving droplet is going to be moving away from that vapor, wet vapor, and coming in contact with more dry gas, thus drying more. And the second thing is these charged droplets, as they're drying, are giving off the charge, thus leading to the development of an electric field, which is live and breathing, a dynamic electric field within the drying chamber that also contributes to displacing that wet vapor with the dry gas, thus allowing for more drying. So there is more drying happening, more efficient drying. At the same time, there is more evaporative cooling because of that vapor, that release of that wet vapor by the droplet. So the solids are always being protected. So if you look at the electrostatic induction, the liquids are moving to the droplet surface, pushing the solids inside. The solids are always protected by the liquids, then by the evaporative cooling. So the theme of the drying process continues where the solids are protected by the liquids at the height of the drying where the temperatures are higher, but as the droplets are drying, the powder particles are moving through the process, through the drying chamber. The chamber is cooling down, the temperatures are going down, the powder is protected. This here is which shows the evaporative cooling process. The chart on the right is conventional spray drying, where in conventional spray drying, when the droplet is sprayed, the solids on the surface of the droplet are immediately dried to form a shell, thus trapping the moisture within the droplet. Right? So the temperatures are high, they continue to dry, those need to be hot enough to dry the moisture trapped within that droplet, that partially dried droplet with the shell inside, the moisture inside that. With the electrostatic drying on the left, what you see is that the solids are inside the droplet, while the blue layer is the evaporative cooling that you're seeing as the droplet is drying. So at the end of conventional spray drying, the outlet temperature is high because you're needing that high temperatures during high drying temperatures to dry the moisture trapped inside the droplet. For electrostatic drying, you can continue that efficient drying at lower temperatures, but still maintain your outlet temperatures low, so your solids are protected from that high temperatures. A visual representation of what's going on inside the dryer in electrostatic drying. The cyclonic flow is important because it allows for more residence time for the droplets to allow for that drying to happen more efficiently. Even though I'm saying more time, the entire process takes place in less than seconds. So as soon as the liquid droplets are formed, you start seeing powder being collected at the bottom of the dryer. Quickly, advantages over conventional spray drying. It is moderate temperature drying compared to the very high temperatures in conventional spray drying, especially at production scale. You might be able to dry at lower temperature at R&D scale using conventional spray drying as well, but you wouldn't be able to replicate the outlet temperatures at production scale. Using electrostatic drying, the outlet temperatures you can see are between 35 to 100 C, maintaining that protection and thus not causing denaturing or denaturation of your material. Advantages over freeze drying, the main advantage is that it's continuous process. You get that advantage of preserving the integrity of your molecule, the same that you get in freeze drying. You get the same with electrostatic drying, and also a discrete particle formation that you would see in spray drying. You see that using electrostatic drying also, that you don't see in lyophilization. You would need to do a post-process, which might not be a thing with drug substance, but if you have a formulation that you're trying to dry, that can be an advantage. I have a few case studies that some of them demonstrate the phenomena that I have been explaining and some advantages over other conventional drying processes. So the first of them is a nanoparticle encapsulation study that we did with University of Chicago and with UC Berkeley. So what we have here is a silicon nanoparticle encapsulation with carboxymethylcellulose. Silicon nanoparticle is a porous nanoparticle that is used to demonstrate encapsulation. CMC it's a surrogate for demonstrating encapsulation. When spray drying is used, the carboxymethylcellulose fills in those pores. But we demonstrated that when voltage is used, when electrostatics is used, a uniformity in coating was observed for the nanoparticle, meaning encapsulation was more superior. So the image on the left is the nanoparticle coated with CMC without the electrostatics. Spray drying without the electrostatics. on the electrostatic dryer. And the image on the right is drying using the electrostatics. So you'd see the encapsulation is more efficient. CMC has encapsulated the nanoparticle more efficiently. Now, this is a surrogate that we have used to demonstrate this, but if you take this knowledge and translate it to a complex formulation, for example, a LNP formulation, you have now what you are using a lipid nanoparticle that is ionizable in a polar solvent. And you have a stabilizer that is nonpolar sugar. So you're drying it, you're pulling the polar solvent out, so you're pushing the ionizable lipid inside. You can normalize, standardize the electrostatics to pull the ionizable lipid inside, push the polar solvent out to dry faster, thus coating the ionizable lipid with your nonpolar sugar, making sure the stabilizing agent is coating the nanoparticle in a more efficient way. That's an application that we are trying to show with this surrogate material. Another example for encapsulation, electrostatic drying has been extremely successful in the field of microbiome and agricultural bacteria. The bacteria get more efficient encapsulation. Here we demonstrated using lactobacillus, which have a pili, which show affinity towards the charge. And the way we show it is when we see viability recovery and shelf life. So on the right, you see when we use electrostatic drying with simple sugar and the bacteria ended up on the surface of the particle, thus being exposed to the outlet temperatures. There was dead bacteria on that surface. So the recovery was lower. When we applied voltage, all the bacteria was pushed to the inside, so thus protected. There was higher viability recovered and more shelf life. So it makes for good visuals. So we have this here. It's not an application that's appropriate for this audience, but we think that this is a good visualization to show the encapsulation effect. Another advantage using electrostatic dryer is with conventional spray drying, with the secondary drying, there is a loss of those fine particles. So for some applications where particle size is smaller, for recovery, yields is lower in conventional spray drying unless you use additional techniques to get higher yields. Using electrostatic drying, because there is no secondary drying involved, the recovery of smaller size particles is much, much higher. The collection yield tends to be about 90%, regardless of what your solids loading is on the dryer. We have recently concluded a trial with filling technique company, Hara-Hofleger. They are dosing technology experts. We conducted this trial for injectables, for filling vials to check for high accuracy in dosing in multiple weights for vials. There was high accuracy in dosing observed during filling trials for various powders and various powders that was generated using electrostatic drying. These results will be published very soon. We just concluded, so unfortunately I could not use the results here. So the results would be published. If any of you is going to attend TIDES in Europe, you will be seeing those results there. So going to the TIDES portion, I have two case studies on oligonucleotides and peptides. The first is oligonucleotides. We conducted a collaboration with CPI in UK on electrostatic drying with oligonucleotides and 18 mer. This slide here shows the particles that were generated, and the purity was maintained from the original material that was dried at a temperature of 75, inlet drying temperature of 75 and 90. The product temperature or the outlet temperature was maintained at 32 and 37 C. The purity was maintained. It was not changed at all, showing that the drying process did not affect the activity of the oligonucleotide. The goal was to show that there was good handling and flow properties, and during dissolution there was no gelling. So those videos show that there's good flowing in the powders and no gelling seen. We have done some extensive work using peptides and solvent. For peptides, one of the advantages using electrostatic drying is most commonly the upstream to drying is the peptides from synthesis end up in solvent mixtures. Electrostatic drying machines come in both aqueous forms, and or you can process both aqueous solutions and also solvent solutions directly. So meaning if, for example, the peptide is in a mixture of, say, acetonitrile and water, the peptide can be dried directly on the electrostatic dryer, so eliminating a concentration or an evaporation step and a post-drying for particle step. So thus you're eliminating 2 steps, possibly removing the solvent and the milling step. So you directly dry the peptide in solvent to get dry powder. Because it's a single-step solvent removal, we also demonstrated that using electrostatic drying, the residual solvents in the powders can be below those requirements set by ICH. We did this using ACN as our example and BSA as a surrogate. So we showed that regardless of R&D to scale up to production and temperature, the residual ACN in the powders remained below those requirements of 410 ppm set by ICH, way below in the final powders. And at the same time, when we took the powders to scale up, the yields stayed above 90. We did the yield studies using surrogate materials of BSA and maltodextrin, BSA being a sticky material and maltodextrin being not sticky, to demonstrate that regardless, we can produce high yields. But keep in mind that yield is a function of time. These were studies conducted in a single business day, so 4 to 6 hours was our study. The longer you run, the longer the yields. Another study we have ongoing at the moment is with University of Bologna. Professor Carbri, they have identified several amino acids that are known to cause impurities, thermal distress impurities during drying processes. So what they have done is synthesize short sequence peptides with these critical amino acids known to cause impurities and process using or dried using electrostatic drying process. And those are the impurities that can be caused and to avoid. And once processed through electrostatic drying, they have shown that none of those impurities were shown to be caused, because this goes back to the theory that I was trying to explain, that the peptide is constantly protected by that evaporative cooling, so no impurities are caused. The electrostatic dryers The machines are available from bench top to production scale. Those numbers that you see are representative of the maximum evaporation capacity of kilogram per hour of liquid that's evaporated at maximum conditions. That's machine operating at maximum conditions of temperature, flow rate, and such. So you would need to optimize for your specific materials to get that out. But that means like 1 kilogram, model 1 is 1 kilogram of water at maximum conditions. I'll briefly a little about our company. Fluid Air is a division of Spraying Systems Co. Spraying Systems is the world leader in spray nozzle technology. Fluid Air has about 40 years of experience in solid dosage manufacturing equipment and pharmaceutical companies. All of our manufacturing is located in Illinois, US, and we are currently focused on the polar dry electrostatic drying at this point. So with that, I conclude. I can answer any questions if you have. You can email me or my colleague. I also do want to acknowledge my colleague, Catherine Buzicki, who is the actual process expert. If you have any questions on process, I am the subject matter, but she can answer any process questions.